Good afternoon, all of you. The afternoon session will start. Okay, now I invite Dr. J. John Wilson to introduce our chief guest, Dr. J. Muthukumar, Bharatiya University, Coimbatore.
Good afternoon to all. I am happy to introduce today's guest speaker, the last session of National Seminar on Emerging Trends in Microbiology. Dr. T. Muthukumar sir, Associate Professor, Department of Botany, Bharatiya University, Coimbatore. An eminent scientist from Bharatiya University, Dr. T. Muthukumar having a teaching experience of 20 years and 33 years of research experiences. He is the awardee of CSR, SFR and SRF and Research Association. He is a university rank holder and cleared 8 exam. He is the visiting scientist of Chinese Academy of Science, China. He completed 2 research projects from UGC and DBT to the tune of 80 lakhs. He is the rating of 7 PhD and completed 6 doctoral students, 14 MPhil scholars. He is a citation of 1914 with the H index of 23. 109 publication in the Tutor journals, 23 GenBank submissions, 23 book chapters. He is acted as a reviewer for more than 132 journals, academic editor and editorial board member in the Tutor journals. Dr. T. Muthukumar is having more number of membership in professional bodies like American Quant Society. So in the short introduction about today's chief guest, Dr. T. Muthukumar sir. So please, you can start this discussion. Thank you, sir.
So good afternoon all. Can you hear me?
It's audible.
Yeah. So first of all, I should thank the convener of this national seminar on emerging trends in microbiological research, Professor Ponmanikam, as well as other organizing committee members, right, for giving me an opportunity to interact with you during this national seminar, actually, right. So today I will be talking about this. I was given asked to talk about this writing of thesis as well as the ethics in science, actually. So today we will be seeing some of these aspects, actually. So mostly I will be sharing my own experience over the years, actually, in these subjects, and I think this will be useful for young researchers, actually, who is new or actually who are pursuing the research in the field of microbiology as well as in the other fields, actually. So first of all, let me share my slides, actually, and then we will go into the lecture. So are you able to see the screen?
Yes, sir. Yes, sir.
Yeah. So today we'll be dealing about this first part. Actually, this lecture consists of 2 parts. Actually, the first part is about the thesis writing, and second one is the ethics, that is the scientific ethics in publishing, actually, right? So let us see. First of all, we'll take up the thesis writing, actually, because it is one of the important aspects. Because today, most of the institutions, they tend to, or the academic institutions, they tend to have this writing of a dissertation or a thesis as a part of a curriculum, actually, right? So it starts almost in the undergraduate in many of the institutions, whereas in others, actually, you tend to have your postgraduate dissertations to be submitted as a part of your degree. And when you are doing your research, actually, it is almost a thesis has to be submitted for the completion of your research degree, actually. So this is how actually this thesis actually or the dissertation happens, actually. So if we take up the definition for a thesis or a dissertation, actually it is a document which is submitted in support of the candidature for an academic degree, actually. So it could be some degree as a part of your curriculum, or it could be a higher degree, actually, or a professional qualification presenting the author's research and findings is what we call it as a thesis, actually. So the presenting of the thesis or writing of the thesis differs from country to country. Actually, it is not universally same, actually. It tends to vary, actually, right? So it might be useful to think about the, always when you're writing your thesis or dissertation, one important thing you should always have in your mind is that the ultimate goal of a thesis or a dissertation is not only for getting a degree. So the finding should be communicated to the scientific world actually, so to the publications actually. So that should be in your mind whenever you are preparing a thesis actually, right? So understand one important thing is that previously people used to complete their thesis before starting writing the research papers actually. But today in many of the universities and the institutions, they have made it compulsory at least to have a few papers published from a few articles published from your thesis work actually, or the research work before you could submit the thesis. So it almost we can say that actually sometimes publication comes first in some of the institutions, whereas in many institutions actually they don't they are not very strict on the publications prior to the submission of the thesis. Right. So there are two terms that are generally used for the research thing that are generally being submitted actually. One is actually the thesis and another is what we call the dissertation. So there is a lot of confusion among people, or the thesis and dissertation are the same actually, right? No, actually there is a difference between a thesis and a dissertation. So it again varies between the type of system which we normally follow actually. So I will tell you about that actually. The biggest difference comes from the purpose for which you actually are submitting the thesis or dissertation, right? So a thesis generally in the Western world is required to obtain a master's degree actually, right? So it is mostly concerned about the testing of the student's understanding in a particular field which he or she has chosen actually. So that is what they consider to be a thesis that is being submitted as a part of their master's degree, whereas the dissertation is actually considered to be a doctoral degree, which generally mostly consists of or focuses on actually what we call as the original research actually. But in nations or countries where actually the academic system is based on the British higher education system, so normally including India actually, we consider dissertation to the work of an undergraduate or the master's degree program, and thesis we refer to the body of the work which is the work that is being submitted at the end of your PhD program. So here you could see there is a large amount of variation between the work that is being submitted or the form of the work that is being submitted at various levels of various degrees actually. So the Western world normally it is actually the thesis is actually the master's degree program, whereas the dissertation is actually the doctoral degree program, whereas in India as well as in other British system following countries, it is actually the dissertation is generally the undergraduate or the master or the postgraduate level degree research that is being submitted, as well as the thesis is actually the PhD work that is being submitted. So this we should always understand. So again, there is also the quantum of the work in the thesis. So we can now consider thesis to be something that is mostly concerned with what we call the PhD program actually. So whenever we talk about the thesis, actually there are two systems that are generally there actually. One is what we call the American system, and another is what we call the British. So in the American system, what happens is that you tend to have a large amount of coursework actually. Coursework means you have to clear a large number of papers, right? And there is a small part is actually on the work that is being, or the research work that is submitted is not very elaborate, right? So it is what something a new findings or a few findings is enough for a PhD degree in the American system. Whereas when you take a British system, actually what happens here is that the coursework is very minimum, but what happens is that the experimental or the research work is very elaborate actually. So these are the two main differences you find between these two systems actually, right? So in India normally we follow the British system. That is why we have a coursework, very small coursework of I think there will be 2 or 3 papers actually you should complete before. That is what we consider to be a part 1 program of the PhD. And second is actually with the thesis actually. So normally we used to have an elaborate research work on which is generally submitted actually. Right. So whenever you take up a thesis or a dissertation writers actually, so these people tends to have because understand one important thing is that so scientific writing is only a skill that has to be practiced actually continuously. So what happens is that with the course of time actually, you tend to have a large number of experience on how to present as well as how to put forward your ideas actually. So that is one of the important things that we normally have, right, with the advancement of the years as well as the experience. So if you take up these people who are writing especially the scholars or the students who are writing the dissertation or the thesis. So these people are generally not very skilled actually. They tend to have very less practice in scientific writings normally actually, right? So again, what happens is that you should also understand one important thing is that previously actually the dissertations was submitted, which we normally submit to or the thesis which we submit to the universities actually. So they tend to have a very small audience, but not now actually, because most of the theses are generally published online actually. So automatically now we tend to have a larger audience. But previously it was generally restricted to the libraries actually, university libraries, institutional libraries. So automatically a very small number of audience of the people who are interested in this particular topic will be referring to this actually the thesis actually, right? And we should also understand one important thing is that most of the theses normally of the dissertations that are normally submitted are generally insecure and mostly they are very defensive actually. So that is why you can always see actually you could always see a large number of the discussion portion will be very massive when you compare to that of the research portion actually, right? So again you also justify actually with a large amount of documents actually, right? And the focus is also very narrow actually. So normally you take up a research problem that is not a very broad one actually. So normally we tend to focus and in-depth studies would normally be done on this type of field that has been generally chosen actually. And one important thing is that, so normally as you write actually, so you tend to develop the skill of writing actually. Every individual person tends to have their own style of writing. So this is one of the important things actually later on I will be showing in one of the scientific ethics, how this generally plays a very important role actually, right? So if you take up this thesis or dissertation normally, this consists of 3 parts actually. So what we call as the front matter, the body as well as the back matter. So let us see what these 3 portions are actually. So the front matter is generally the title page actually, where you present the thesis or the dissertation title actually. And second comes the approval page. This is what there will be a few certificates actually. That is what we call it as the approval pages actually. And then comes the acknowledgement. So you acknowledge the funding agencies, you acknowledge the persons who have helped you actually. So all those things comes under the same funding agencies and other things under these acknowledgements actually. Then comes the table of contents or the content, and in certain cases we also present the list of tables and figures that are being incorporated into the thesis actually. And finally we tend to have an abstract that is actually what we call it as a summary actually. In Indian system we call it as a summary. So in the European system they tend to put it at the front and they call it as an abstract actually, right? So when we call it the body of the thesis actually, the body of the thesis or the dissertation consists of 6 parts actually, right? So it starts with an introduction, right? So then comes the review of literature, then comes the materials and methods, results and discussion, and finally we get the conclusion of the summary. So this is what we call the IM or the pattern of actually writing a thesis actually, right? And the back matter mostly consists of the list of references and as well as the appendix. So you may, in certain type of studies normally we may be using certain questionnaires that will be used, right? Or certain data data will be generated actually. So all these are generated at the end of the thesis. That is what we call it various appendix, right? So let us see if each and every part and what are their importance actually. So introduction actually is a very important thing because this is where actually you introduce the subject what you are generally presenting. So this is one of the important aspects. So normally an introduction should start with a wider thinking actually, or wider or you give a wider scope for the scope of this problem which you have chosen actually. And normally you narrow it down to the objectives of the hypothesis which you are generally generated. So this actually consists of, this introduction should always consist of certain parts, important parts actually. So that is what we call it as the statement which generally deals with which generally reveals the problem. So why this? What is the problem and why it has to be solved actually? So you might by through the review collection and other things actually going through the review literature actually. So you might have got an idea. So you might have seen that there could be some lacunas in certain areas, and you wanted to plug these lacunas, or you have to show, or you have to further, or you have to confirm many of the things that have been unsolved in the previous studies actually. So all these things that should be actually shown in the introduction actually, right? So then there should be a brief overview of the study actually, that's what generally studied. So what is the hypothesis or the proposal that is being proposed, right? And in what ways this could be generally could be solved? For example, how are we going to collect the data and analyze the data? And what are the limitation? Understanding the limitations of your study is also a very, very important aspect. You should keep it in mind. So actually you should always understand the end of your research is the starting point for the next research. So this is what should always be kept in mind actually. So you start with something actually, and you try to find a solution actually. But at the end of the study, you could see that this particular study would be revealing a more amount of more studies actually, or more ideas actually. So which needs further studies actually. So that is what we normally say. Any research is not an end actually. So it is only the starting point of the next research actually, right? And there should also be a discussion about the significance of the study. That is, who is going to benefit from the studies actually, or the findings actually, right? So what is the study is going to be what is the advancement of the study over the already known or the proven aspects actually. So again, the next important part of your thesis or dissertation is actually, that is what we call the review of literature actually, or literature review actually. So there are certain key points we should always keep it in mind actually, right? So you should always compare how the previous work, right, or the previous findings of the hypothesis that has already been proved or disproved actually, or relevant to the present study, right? So again, you could also argue about the agreement or disagreement between with the different authors actually, right? So you have to also mention the relationship of the particular what you have done is actually with the previous work, and how it is relevant to the area of study actually. And you also try to find out the gaps that is also already present in the knowledge that is available in that particular field actually. And you also recommend whether further studies or the research is generally needed. So understand one important thing is that when you make a very good review of your subject actually, so automatically this would come up or bring out one or two good actually review papers. So understand one important thing. In many of the journals you might have seen the review articles actually that is being submitted. So many of these review articles are actually done by this actually research students actually. So when you start to read and understand the previous studies of the literature that are already available, this could bring in a very good what we call it as a review article or the review paper, right? So next comes the portion that is what we call as the methods actually. So it is actually there are this is how you what is your experimental design, how you collect the samples and how you transport the samples, and what are the various methods that are being followed, and all these things actually come to this section that is what we call as the methods of the materials and methods actually, right? So here you should always understand one important thing is that, so you should always have it in your mind the research questions or the hypothesis that you have formulated, and you have here what you do is that you mention in the methodologies how you are going to solve this hypothesis or solve these questions actually, right? And one important thing is that you should also mention the rationale begins selecting certain methodologies that is going to be followed actually, because for a particular analysis for a particular parameter there could be several procedures that would be available actually, right? So understand one important thing, it is always important to understand the recent procedures that is generally being used. For example, if you take up microbiology, so previously people were using, for example, people were using what we call it as the morphological as well as the biochemical analysis as a parameter or identification criteria for many of the bacteria or microorganisms. But today in modern microbiology we don't accept those things actually. So only molecular characterization is generally being accepted, right? So you should always understand such type of things also, because at the end actually when you are going for a publication, such type of limitations would always hinder the publication of your work in a very good journal. So whenever you are writing your thesis or you are proceeding with your work or the thesis work or a PhD program or your MSc program, you should always keep this in mind, that is ultimate goal is to bring up one or more number of good papers in good scientific journals actually, right? So even if you take up the data collection, there are different types of data collections. For example, sometimes it can be observational data actually, or sometimes it could be experimental, or sometimes it could be simulation, or sometimes it could be a derived data. So it depends upon the type of experience or the procedures which you are generally following, right? And another important thing, whatever we see is that, so whenever in these materials and methods, it is not important to describe already well-known established methods actually. So unless you do certain modifications, it is not necessary to have the entire methods actually, right, or elaborate the methods that is generally being followed. For example, in large number of theses and other things we normally see, so some well-known procedures will be repeated or will be explained actually. That is not necessary actually. So just you mentioned, unless you made some modifications. So if for the existing procedures you have made some modifications, so just you mention only the modification, or just give the or cite the references actually, right? And another important thing is that whatever the statistical analysis which you have done in analyzing the results should also be included in this particular section. That should be the end of the methodological section actually, right? And generally when we speak of this methodology, there are just 4 things you should always keep it in mind. So what was done to answer the research questions, the one. Second is to describe how it was done, right? To justify the experiment will be said, right? And finally, you have to show, right, how the methods or the results were generally analyzed. So that is what actually the statistical procedures and other things. So this is actually what we call it as the methodology section. And next comes actually the important section, that is actually the results, which is actually your findings, right, on the study actually, right? So you should always understand one important thing is that the majority of the theory should always be written in the past tense. For example, if you take up the materials and methods as well as the results, because all these things should be written in past tense actually. Because already when you are presenting already the study is generally being completed and you are presenting only the completed study actually, right? So again, every research must be also have a method set out in the methods section. So whatever you are showing in the results, because we see at many times what people do is that they used to have a large number of parameters, or they used to present large number of parameters in the results. But the methods or the procedures for evaluating these parameters, you don't find them in what we call it as the methodological section. You should always see that this never happens actually, right? So again, you should always understand one important thing is that, so whether it is the positive or the negative results should generally be presented actually. So people always have this actually, one idea is that, so whenever some unexpected result comes actually, they don't present that particular results actually. So whenever sometimes what happens is that your results may be deviating from the normal thing that is generally present or generally being reported actually. So such type of things actually, because understand one important thing is that in biology, right, and biological sciences, you tend to have large number of variations, right? So depending upon various conditions actually, or varying conditions actually. So it is not only that only the positive results should be present, it is not like that actually. Sometimes the negative results could also be of significance in science actually, right? So again, the results should always be arranged in a logical sequence actually, right? And you should never discuss any portion in the results in this particular section. So because we could see certain people discussing certain things in the results section. So that when it is a separate section, automatically it should never be done in this results section, right? And one important thing is that presentation of the data. So with the presentation of the data is normally done in either the graphical forms or in what we call it as the tabular form. But certain people, what they do is that just to increase the page, what they used to do is that they tend to present the data both in the form of figures and tables. So this should be avoided. And another important thing in the results is that we should never repeat the data that is already presented in the tables and figures actually in the results section. So what we used to tell is, do is that, so this parameter has, for example, this centimeter. So all these results will always be presented in the tables or from the graphs, which an examiner or a audience could easily see, or when they see the table or the figure. It is not necessary to repeat it again in the results actually. And one important thing is that the presentation of the data is very, very important actually. So normally when you are writing preparing a dissertation or a thesis, the first important thing is that you have to finalize the data, that is actually the tables and figures first before writing the results actually. right? So again, understand the presentation of the data is the only a very important as well as a difficult part, because people often tend to have a large amount of confusion. So what are the data that has to be presented in the form of figures, and what are the data that has to be presented in the form of tables, right? So understand one important thing is that figures are very easy to understand compared to that of the tables actually. You understand? So all the trends and other things would be very easily seen when you look at the figure. Whereas the tables, it could be that in the tables actually when you present the data, automatically what happens is that one has to read through the tables actually to understand what is actually there, or the data, what is actually there in it actually. Right? So there is no what we call the thumb rule, which is right and which is wrong actually. So it depends upon the person actually and how to present this. And one important thing you should always take it in mind is that whenever you are presenting the data in the form of a table, so it is very important to have what we call it as the table, the data or the values, which are a larger number of values. So decimal values should not be presented in the form of tables, because when you are presenting something 0.005 or 0.007 actually, so it will be very difficult for a person actually to read through these values actually. Can you understand? So such type of fractional data or decimal data could easily be presented in the form of a figure actually. So automatically the variations of the trends could easily be formed by the audience or the readers actually. Right. So understand one important thing is that whenever we talk of a figure actually, so the legend of the figure will always be presented as the base actually. So the figures are normally read from the bottom to the top. It is not from like a table which we normally read from the top to bottom. It is actually from bottom to the top actually. Right. So normally understand the figures, there is a large amount of variation in presenting the figure. So you should always see that we can, when sometimes a more number of figures for a particular experiment is available, or the similar parameters or interrelated parameters are available automatically, this could be combined into a single figure actually, right? So understand one important thing is that each individual figures or the images in a figure should be labeled actually. So this is also one of the important things, and this should also be mentioned in the legend section, right? So this is very easy. I think many of you might be familiar with this actually. So it is always when you are presenting the common figures automatically, so each figure should be differentiated, and this differentiation should also be reflected in your results section actually. So whenever you are referring to a figure actually, for example, when it is a multiple figure, so you always note which actually of the images is generally being referred to. For example, if it is a figure 2A, that should be included in your results section, right? And one important thing just I wanted to tell you about this images, that's micro photographs or any type of photographic images in biological science, is that you should always include a scale in it actually. So this is what many of the people don't do actually. So just they give the magnification actually. So magnification is the not way of representing the value, the scale of the or proportion of the image actually. So what happens is that, so whenever you are presenting a micro photograph or a photographic image actually, so it is always important to introduce a scale actually, right? So when you are presenting it in the form of a graph actually, it is always understand one important thing is that the main variables that are generally being measured is generally should be presented on the y-axis, and the categories that are presented are generally presented on the x-axis. So normally you tend to have large, you tend to vary the various types of factors or parameters are generally distincted or differentiated by different types of colors or patterns actually. So today in graphical presentations there are large amount of facilities that are available where you can use large amount of these variations to show in these images actually. So these are some of the common type of figures that are normally being used in the presentation. So it could be a bar chart actually, or it could be a bar chart combined with a line graph actually, or the line graph actually. So sometimes it could also be a box plot, right, or principal component analysis or the regression analysis, right? So there are different ways in which actually different types of graphs that are generally being presented. So depending upon your nature of the data, and how it's generally being presented. So sometimes you also tend to break the scales actually. So here I have shown you one example is that. So whenever you tend to have a few values that is exceeding the others actually, or there is a large variance, there is not much variations in the data actually. So there is one way of making the distinction very clear is to cut the axis actually. So this is one of the things that is usually being followed. And understand one important thing is that the correct way of presenting the data is always one of the important things is that. So sometimes understand there are two types of data, that is one is what we call the continuous data and another is what we call the discrete data. Understand one important thing is discrete data should never be represented by line graphs actually. So it should be only be represented by bar charts actually, whereas continuous data could always be presented on a line graphs actually. So this distinction should this character presentation should always be understood, because in many of the theses as well as in the research articles what we come with, so for many of these discontinuous data or the discrete data, they tend to use the line graphs. So that should always be avoided actually. Right, so again the tables is also another form of presenting your data actually. Understand one important thing is that tables are very, very important, because sometimes in your publications or in your thesis, sometimes people used to take up your data and they used to do the meta-analysis. So when you are doing such certain type of meta-analysis on certain aspects actually, so only the figures would not reveal much of the what we call this much of the information, right? So whereas the tables are very, very informative from this point of view. So whenever you wanted your data to be reanalyzed or used in further studies by yourself or by others actually, presentation in the form of tables are very, very essential. So you know the table consists of rows and columns actually, and the portion or the cells actually actually contains the values actually. Right? And another important thing is that apart from the title, the footnotes are also very, very important. So whatever the abbreviations that are generally used in the tables actually on the column headings or on the row headings actually, so this should be explained at the bottom in the legends actually. And also various other statistical presentations that are generally, for example, you might have used that mean separations and other things. So whenever you use this mean separations, or sometimes you might have presented the error bars actually, so errors actually. So it could be standard deviation or standard error, and all these things should be provided in the legends actually. So this is how tables are generally being formulated, right? And the last portion regarding the body of your thesis is generally considered to be the penultimate thing is what we call the discussion. So you have generated a large amount of data, right? And you have presented a large number of data. And one important thing is that you should always understand, right? So how this research or the prop or the study you have conducted actually finds actually like what is already known in the research, right? And how it is differs, and what is the further understanding it generally brings about, right, to the existing knowledge. So all these things will be generally included in the discussion. So whatever when you are doing in the discussion is that very, very simple. What I ask my students or anybody to follow is that, so you take up the key results actually from your thing. Just you write down the key results. So leave some space actually and write down the second key result. So you go on writing almost all your results actually. And for each key result actually, so you see how this results actually agrees or disagrees with the previous information of the study that are already available, right? So next what you do is that you also you also write down or you also discuss the reasons for this variation. So either it agrees or disagrees. So what is the reason for it, right? So for each and every result actually, you write a discussion individually, and finally you remove all the results automatically. Your discussion will almost cover all the results, and just when you go through it and you correct it actually, automatically a perfect discussion of the results would be. So sometimes what happens is that many people what they do is that they don't do it this way. So what they do is that some of the key results may be missed in the discussion part. So once you do like this actually, automatically all the results would enter into the discussion section, or the key results actually. So it is not necessary that every result should be discussed actually. So how far your hypothesis, what are the important things for your hypothesis or for your objectives, that should be discussed elaborately in the discussion section, right? And one thing important thing is that you always try to interpret and explain the result. That is very, very important, right? So how far your results are answering the research questions, and that could be that should be discussed actually. As per then actually the justification of the approach of the particular study is also very important, right? So already previous studies in that line might be there actually. So how far your study is actually superior to the previous thing, that should also be noted in the discussion section actually. And it is very important to critically evaluate the study and keep it in mind actually. The discussion section should not exceed the sum of all the other sections. So this is sometimes we see it in the what in the master's program presentations actually, right? So what the students used to do is that they used to have a discussion for 20 or 23 pages, and all the other sections would be either 10 to 12 pages or 13 pages. So that should not be that. So whenever you are writing your discussion section, you see that it does not exceed the other sum of the other sections actually, right? And the more emphasis is that the importance of the results should be emphasized. Don't repeat the results in the discussion. So this is also very important. So do not repeat the results actually, but you always explain the importance of the results actually. And another important thing should always be understandable about the shortcomings of the study, because understand every science is like two sides of a coin actually, right? So when there are certain advantages, there could also be certain disadvantages. So this shortcomings of the study should also be mentioned in the discussion section. And finally, the concluding part of the discussion section should always direct towards the future actually. So what are the future directions that should be given? And one important thing is don't go on reiterating your results. That is also very important, as I already told you. So don't repeat the results or go on repeating the results in the discussion section actually. And the final part of the body is actually what we call the summary. So here what you do is that you sum up all their major results actually, right? And you also provide your recommendations and suggestions in this particular section. Recommendation always should include what should be done after the research actually, and it should be precise actually, right? So the summary should sum up the entire work actually, right? So if your study has involved certain questionnaires or statistical tools actually, that should also be included actually, right? And one should always be very creative when you are writing your summary, right? So instead of so what they do is that is what in other countries what they used to do is that they used to have an abstract at the front actually. So when you study the abstract or the summary, one should be clear of what is your research problem, how it is done, and what are the key findings of your study. That should be easily be understood when people are studying or going through your summary, right? Or sometimes graphical presentations could also be given in the form of could be used in the when you are recommending actually. So these are not normally followed in Indian system actually, but in other systems they generally follow. So this is how actually the body of the thesis of the dissertation happens. And finally, the important section and which many people don't take much care is actually the reference section. Understand one important thing. You always understand that many of the papers gets rejected because of these references actually. So either there could be wrong references that are being cited, or they are not actually cited correctly actually. So references are only important thing. This section should be taken much care when you are preparing your thesis or dissertation, right? So again, it is a complete list of sources that are generally formatted is what we call it as the references actually. And there are different ways of actually presenting the references actually. Understand whatever the system or the style you generally follow for citing or listing the references, you be uniform throughout actually. You can understand. So when you use an abbreviation for a journal name, use the abbreviation for all. Just having 2 or 3 references, the whole journal name, and then you don't actually abbreviate it. So when you are using an abbreviation, you abbreviate it uniformly throughout. So whenever you are abbreviating, you see that there are standard abbreviations for the journals actually, and strictly follow the standard abbreviations. So many people they get to abbreviate the journal names by themselves actually. That is not the right way of doing it actually, right? So when there are same type of same group of persons actually have published more than one papers actually in a particular year actually. So normally these are differentiated by the alphabets like A, B, C at the end of the years actually that is being mentioned. right? And for research papers, actually, it is actually the author's name, right? Author or author's name, actually. So when there are more than two authors in a study, actually, when you're citing in the text, actually, normally you use et al, actually, right? And in the reference list, actually, normally all the authors are designated, but in certain cases when the authors are more in number, actually, so certain studies may contain 50 or 60 authors. In certain cases, et al can be used after fifth or sixth author name, actually. So it depends upon the style, actually, because many of the institutions or the universities, they don't have a strict, actually, what we call it as a format for citing or listing their references, actually. So again, the books also is very, very important. So whenever you are citing the books, actually, you see that actually the editions are rightly being actually cited, actually. So sometimes what happens is that they don't cite the editions, actually, and it is also very important to name the publication that is the press, actually, as well as the place of publication is also very, very important. So when you are citing the chapters, actually, in the books, actually, so automatically one important thing is that the editor should always be mentioned, actually, and similarly the page range should also be mentioned apart from the book, actually, right? And when you are citing the online journals, it is very, very important to mention the DOIs, actually. And normally try to avoid citing the thesis, actually. So now it is not a problem because many of the theses are available online, actually, so that is not a problem. But previously this was a problem because many people might have not seen many of the theses which they normally cited. So that is why we normally say that instead of citing the thesis, actually, it is better to cite the paper that is being published from the thesis, actually, right? So similarly, we can also now, actually, because many of the literature are also available on the websites, actually, or web sources also may provide many of the information. In such cases, normally we should also provide the date of accession, actually. So when this actually the site was accessed, that should also be mentioned, actually, right? So these are some of the things about the writing the thesis, actually. So there are many type of thesis. So what I told is actually the Indian system, what we normally follow, the IMRD, normal thing. And there is also another type of thesis, that is what we call it as a comparative. That is actually what we normally do is that we tend to have a general introduction and a general review of literature, and each and every objective will be presented as a separate paper. So which will be self-contained. That means it will be having an introduction of its own, it will be having a methodology, it will be having a results, and it will be having a discussion. And finally, we will be providing a general, either results will be stopped along with the results, or we used to discuss the results. Or sometimes in certain cases, what we used to do is that we generally have friends to have a common discussion and a common reference list, actually. And there is also another form of thesis, that is what we call the compilation thesis. Here what we do is that they used to publish different types of a number of papers from a study, actually. And what they do is that they used to compile these papers and write what we call the introduction as well as the summary, and all these things are generally being put into a form of a thesis. So normally in many of the Western countries, actually, compilation theses are very, very common, actually, right? So in India also, we use this compilation thesis for one thing, that is for B.Sc and D.Lit, that is Doctoral of Science or Doctoral of Literature. That for both these degrees, actually, normally B.Sc and D.Lit, actually, we use this compilation thesis format, actually. So we tend to have all the publications that is being published, actually, right? And we used to have a introduction as well as the summary, right? And we used to cover each and every chapter, actually, will be a published paper, actually. So these are the various ways in which actually the thesis are generally written, and our dissertations are generally written and submitted. Right. So let us go into the next important thing is what we call it as the second part of our lecture, that is what we call as the scientific ethics, actually. Right. So when we talk about this scientific ethics, because it is very, very important today, because there is a large pressure on the researchers, right? That is as resulted in the introduction of or the following of various types of wrongdoings in science, actually. So that is what is very, very important. So literally, when you say what is ethics, actually, so it is the moral principles and values a civilized society generally follows. So normally what happens is that we understand what is right and what is wrong from our parents, right, from our young age actually. And when you come to school, actually, the teachers and the mentors generally teach us what is right and generally what is wrong. And in certain cases, the religious institutions also plays a very important role, right? So, what is to say what is right and what is wrong actually. So, it is understand one important thing is that, as Albert Camus told actually, a man without ethics is a wild beast loose upon this world actually, right? So, because understand one important thing, scientists always trust the result that is being reported, because they always think that is generally based on sound protocols, right? And the conclusions or what the conclusions that are drawn are generally valid in light of the current knowledge. So what has been people think is that whatever being that is being presented is genuinely being done as well, right? And another important thing is that understand most of the research today is generally being funded, and most of these funds comes from the taxpayers' money or the public money actually. So we always expect the science of the researchers to be most honest and consistent. So yes, anything what happens is that sometimes we always feel that actually researchers should provide the results actually without accurately actually without any bias and any exaggeration. That is what we call as a hyperbole. So exaggeration without any exaggeration, right? So the scientific ethics is always concerned about this honest and integrity of all stages of scientific science practice actually, right? So it is not only from reporting the results, but it's also important in the life to properly articulate the collaborators or the people who has worked in that particular thing. So there is also a large amount of evaluation parameters here today used to actually what we call it as to scale the researchers actually. For example, not only the researchers actually, the journals that are generally being published actually. So for example, there are a large number of parameters like what we call it as the impact factors, H index and citations and other things. And there are also, it is not only peer pressure for actually not only the researchers, but it's also for the publishing companies actually today, right? So later on I'll be discussing about these things actually, right? And it is also, there is a large amount of still competition for publishing in very high impact factor journals actually. So publishing in high impact factor journals are very, very important, because if you want to get grants actually or projects actually, so it is very, very important to publish. You have publications in good journals actually, right? So automatically what happens is that the scientific community is generally under a lot of pressure actually, right? So again, it is not only for getting promotion and going higher, but also for retaining in certain courses. So India, there is not much problem, because once somewhere you get settled actually, that is not a problem. But in the European normally you tend to get only what we call it as you don't get a permanent job actually. So automatically for every 3 or 4 years actually based upon your performance actually, right? So you are just actually you are held or just you are generally relieved from a job actually. So that actually results in large amount of pressure on publishing actually, right? So why do we publish? What is the need for publication? We should always understand, right? So as we had seen in the first part, it could be a partial fulfillment for certain academic degrees. For example, you are a master's program or getting a higher degree, that is a doctoral program actually. And one important thing is to your curriculum or CV actually, or sometimes for promotions, because today we know that actually faculties are under very peer pressure actually, because when going to the next stage, they always request for, or they always ask for certain criteria that has to be satisfied, and one thing is actually the publications actually, right? And certain people also go and publish to get their names in the particular field actually, right? It is also very important to build an academic reputation, and it is also important, as already been seen actually, to get the projects as well as the fellowships. And sometimes it is also used for personal pressure. So very few people actually do this for this personal pressure actually. But many people actually, it is for many other of the previous causes, they do that publications actually. To understand publication is not very easy actually, because your work after writing and communicating has to go through a long process actually, and a very hard process before your paper gets accepted. Right? So there is a large amount of hurdles that is always awaiting your research before it finally gets published. Right. So there is a large amount of scientific misconduct that generally happens actually. So for example, so because of the speed pressure actually, it always makes people to falsify or fabricate the data, right? And they tend to reproduce others' ideas or even the uh, uh, the papers that are being produced. That is what we call as plagiarism, and even sabotage also generally happens. So the scientific breach of ethics happens in different forms actually. One is what we call as the plagiarism. Understand? None of this generally happens in countries where English is not their native language. So that is the biggest problem because whenever we write in a foreign language that has a large amount of limitations, we tend to have a large amount of limitations. I think none of us has English as our mother tongue actually, right? Automatically, we tend to because understand this plagiarism starts from our childhood in India, in some of the countries actually. So because right from our childhood we are always taught to actually reproduce the fact, right, as such actually. Understand? So even in your Tamil, I think that poem should be written actually. Similarly, we have a large amount of definitions and other things actually. And even a person who gets a good mark, or you could see that he reproduces near to what is already available actually, or available in the books actually. So a person who reproduces perfectly would be given the maximum marks actually. So this is what happens. Always the later on stages, what happens is that the people tend to keep it in mind all these things actually. They do the same thing. So when you do the same thing in a research, that creates a large amount of problem actually. Right? Second one is that because there are a large number of pressure actually, right? So automatically people tend to falsify the data actually. So what they do is that without conducting a study actually, they tend to generate data actually. So this is what we call as the falsification of the data. And sometimes what they used to do is that, so understand in science, so all experiments would never end up in success actually. So normally you expect something and perform an experiment. You don't always think that the experiment would always be successful. So there could be a lot of failures before you tend to succeed actually. Can understand? So again, researchers or especially the students, because they have a very short time of 3 to 5 years to complete their PhDs actually. So they are not, they do not have time to actually repeat many of the works actually. So automatically what they do is that they start to adjust or to manipulate the data. So this results is what we call the falsification of the data. And certain amount, another important thing is the redacted or the duplicate publication. So here what you do is that you publish the same papers, so just to increase the number of publications in your CV actually what you do is that you tend to publish the same material or the same content in different journals actually. So that is what we call as the redundant or the duplicate publication. Again, there is also one important thing that is actually you tend to provide conclusions that are actually beyond the scope of that particular study actually, right? So this is generally done for actually getting publicity actually. So for example, with this COVID in the last year, you might have seen there were a large number of publications on this COVID actually, right? So every pupil has to have their name in this particular field actually. So automatically you could see there were a large number of retractions also during the studies, many of the studies that was actually performed on this COVID actually. And another important thing is the gift authorship actually, right? So this is both receiving as well as giving actually. So gift authorship means actually a person, so whenever you are giving an authorship to a person, you should have contributed something to the study, either in the form of designing the experiment, performing the experiment, or preparing the manuscript or anything actually. So certain people actually, because of their sheer status in that particular area actually, they are given actually, they are included as the authorship. For example, in many of the institutions you could see what we call it as the head of the institution or the head of the department would always be a permanent author in almost all the publications that is being published by the particular department or the particular institution, although he has not contributed anything to it actually. So this is what we call as the gift authorship actually, right? And another important thing is that not giving sufficient attention or consideration to the scholars or the postdocs for the nonsense. So this also happens. So whenever persons are working on the projects actually, or students are working on the projects, so whenever the papers gets accepted for presentation in conferences or foreign scientific gatherings or something actually, it is scholars or postdocs are not given the chance actually. It's the investigator generally who takes the credit, right, and goes and presents the paper. So this is what we call it as not giving sufficient attention or the consideration to the scholars actually. And again, self-promotion at the cost of the team members. So here what they do is that this happens in certain institutions actually. So when you are having different departments actually, so the department, the institution head used to collect information from the different departments and he used to publish it in his name actually. So that is also a very important thing that happens in many of the institutions. And treating the juniors actually, or treating the colleagues in a very fluid way actually, right? And another important thing is called as the naturalism, that is actually the contingent or the duplicity, right? So using the power actually to make certain advances in science actually. For example, as I already told you, the institutional head or the department head actually, right? So this type of things is generally what we call the breach of ethics of science actually. So this has resulted in large amount of retractions actually. So there are a large number of papers that are being published, and in due course of the time actually there are also a large number of papers that are being retracted or being withdrawn actually. So you know this last year was a COVID year actually, so there was not much activities in science, especially in the laboratories actually. But even in that time actually you could see there are a large number of retractions, almost more than 1,500 papers actually were retracted from different journals all over the world actually. And these retractions also included some of the papers that was actually published in great medical journals, very important medical journals like what we call the Lancet or the British Medical Journal actually, right? And this retractions actually is even in the time of this non-activity, right, or the research activity, you could find such large amount of retractions of the withdrawing of the papers for various reasons that will be conducted. So if we take up India is we should be proud because we are the top 5 countries in the world actually, which produces the largest number of articles of fields in different fields actually. So normally we are in the 3rd position and almost next to China and the United States actually, right? So, and this you could also see that we also, many of countries except China or the major paper articles publishing countries, most of these studies are from what we call the biological sciences or the health sciences as well as the medical sciences. And except one country, that is actually China, which produces more number of papers in engineering actually, right? And if you take the two giants actually, for example, United States and Japan actually, most of their studies or the research publications are from the health science and the biological sciences actually, right? So in India, there is not much variation between the different types of publications coming from different fields of science actually, right? And we should also consider another thing. India is also one of the highest countries with the retraction rates actually. So because of various reasons, many of the papers published from India are also retracted actually, right? So if you see there was a surge in there was an increase, a sprout increase in the increase of the retraction levels between almost 2004 to almost 2014 actually. And then it started after 2020 to 12 itself, it started to stabilize actually. So because understand this was the period after 2004 to 2012 was the period when many of the journals has moved to online actually. So previously many of the journals had only the hard copies that was being circulated. So normally finding many of these type of scientific thoughts is very difficult in such circumstances. So once these journals went online, many of these what we call it as the scientific misconducts has come into light actually. So that has caused a large number of increase or a sudden sprouted increase between 2004 and 2012 actually. And now almost that number is generally being saturated actually. And you could see the most number of problems or the retractions are based on the studies actually, which has actually mostly is the fabrication or falsification of the data as well as the plagiarism, right? And there are other types of misconducts. And one important thing in recent years that is being surfacing too much is what we call the fake peer review. That means you actually create the false reviewers by yourself actually, or you create actually the communication, especially the email IDs of well-known people, and you tend to review your own paper and submit the review report of your own paper, and then showing you the examples in the coming slides actually, right? And if you see, most of the papers that are being retracted are mostly due to the plagiarism actually, or the duplication of the text actually. And there is also the next term is generally what we call as the flawed image. And you could see after 2010, and you could see another important thing that is surfacing is the fake peer review. So it is one of the biggest problems today we face in the publication sections, publication aspects actually, scientific journals. So now I'll be showing you some of the real-time things that are generally happening. So this is for your just for your information actually, and you could see how this generally happens actually. So this was a book chapter that we published in the year around 2015 actually. So I wanted to you to look at this figure B actually of this figure 3 actually, 3B actually, right? And this is actually a paper that was published around in 2019 actually in a journal actually. And here I wanted to draw your attention of this figure 1C actually. You understand? So the previous one was actually this figure that is was 3B, and figure 1C. What is that? It was the same figure that what we published in 2015 has been reproduced by another group actually. So only thing what they have done is that they have just reoriented the images, right? So once they are reoriented, automatically they thought it tends to become a original figure. It is not so actually, right? So what they have done is that they also removed the scale part of it actually, right? And the same image has been published as their own image actually. Right, so this is not new for us. Already, this is actually a paper from my PhD thesis actually. So it was published in 2000 actually. So here I wanted to draw your attention to this figure C actually of this figure 2, this image C actually. And this was a paper that was published in the year 2005 in another journal actually. So you could see the same image, right, has been reproduced. And only thing is what the particular person has done is actually he has actually reoriented the image. So in the previous case actually it was 90-degree reorientation, and here it is actually 180-degree reorientation. So this is the paper which we published, image which we published, and this was the paper that was published in 2005 actually, right? And once you what you do is that you tilt this image to 180 degrees, automatically you could see it almost perfectly matches with the figure actually that is there actually, right? So this is actually done, and this is what is double submission or double publication. So what people think is that you understand one important thing is that a paper should be communicated to only one journal, and only if you get after getting the decision from your journal actually, right, automatically the paper has to be submitted to another journal. But many people think is that because it takes a long time, right, it takes 3 to 6 months actually. So after waiting it, and when your paper gets rejected, automatically when you again resubmit it to another, so it is lot of waste of time actually. So people think that actually it is very ideal to submit a paper simultaneously to 2 or 3 journals actually. So from the journal which you get a positive response first actually, that could be taken into consideration, and that could be. So this is a very false thing, and this is considered to be a scientific misconduct, and this should never be done. Because understand, certain journals actually when they are what we call it as they don't have papers, they don't ask you actually, or they don't even send proofs actually, especially certain trade journals, they publish the papers actually. So here I am giving you one simple example of a paper actually that has been published in 2 different journals actually, right? So everything is the same, content is the same, authors are the same, the sequence authors are the same, everything is the same. Only thing is that it has been published in a different journal actually, right? So this is considered to be, this is what we call the double submission. So most of the double submission comes from countries where actually papers are published from in languages other than English actually. So what people think is that publishing the paper in their own language, for example in the Chinese language, right, the paper could be, because understand Chinese language has got a very limited audience, because who knows only Chinese actually can go through these Chinese papers actually. So when it comes to an English, when it is the same paper is submitted to an English journal actually, automatically the audience becomes wider actually. So people think that actually to have a wider audience they should publish in English, and for the Chinese actually the English for their own people actually they should publish in Chinese actually. So this is totally a wrong thing. And understand sometimes actually this publication that is being published in a local language is allowed in English actually. but this should be done in the consultation with the editor actually. So that should not be done. So this is one very important example is that actually you might have heard about this discovery of this artemisinin, and actually this person too actually was awarded Nobel Prize in shared the Nobel Prize in 2015 actually. Understand this paper was published in Science actually, and understand this paper was cited only once outside China, right, before the award of this Nobel Prize. So understand some good works generally being published in local languages actually, local journals actually. So what happens is that if such type of works are available actually, they could be published in English for a wider audience. But understand all these things should be taken only after a consultation with the editor of the particular journal, English journal that has to be submitted actually. And this is another thing where we call this what we call plagiarism. So what people do is that they used to take content from different journals actually, and they used to make that paper of their own actually. So as I was telling you, every person has to have a particular style of writing actually. So when this manuscript came for reviewing, just when I was going through this manuscript, I could see there was large amount of change in the style of writing actually. Because understand, throughout a manuscript and a person or a group generally prefers authentication, there will be a single style of writing. But when we go through such type of papers, you could see the style of writing generally changes from section to section. So when we explored this, what the problem was there, and we could see almost, we could see here, so the same thing, the same paragraph was taken here for the same paragraph, and almost you could see the three colors actually. These three colors represent three different journals actually, and all this content that was taken from three different journals, this person has made it his own paper. So here you could see it was from phytochemistry, right, and it was from food chemistry, and it was from Journal of Cereal Science. So all these three papers he has taken, right, and he has made a review of his own actually, right. And this also happens in certain journals, which, for example, taxonomical descriptions actually. What people discuss the taxonomical descriptions never change. For example, this the left that I have shown you here is actually the manuscript that was the description that was presented in a manuscript, and on the right is actually the original description. So here you could see almost nothing has changed, including the dimensions of the structures actually. You understand? So the manuscript was submitted from a group in Goa actually, right? So here you could see this particular original description was based on the specimens that was collected from USA. So consider, so the specimens considered from USA as well as from Goa of India will not have the same dimensions actually. You understand? So here you could see it is almost the verbatim same of the original description. So this is also should never be done actually. The description should be based on your own observations and your own studies actually. And again, there is also another type of work that is being done actually, where to increase the number of publications. What we do is that you prepare, this was a manuscript actually that was received actually, right? And here you could see the work was conducted for 2 years actually, 2006 to 2007 actually. So what happened was that when we are searching the literature for this for papers published by this particular group actually, so I could see that there were 2 papers already published by the same group actually. Group means actually that there was a single author, but the 2 other papers that was previously published, right, pertains to 2 different years actually, 2006 and 2007 actually. But it had a large number of co-authors actually. Both these papers had a large number of co-authors. But the previous paper that was submitted to a journal had only a single author actually. You understand? So here you could see there is also a large number of data was also the same actually, right? And finally what happened was that actually this paper, it was to increase the number of papers actually, and after searching the person in Google actually, I found that actually he was a director of an institute actually. So what he has done is just to increase the number of papers, he has combinedly published the 2 years work, separately actually he has published it, and he has combined the 2 years data into a single paper, right? So this type of publications would almost this type of scientific misconduct would result in one is what we call the retraction of the article or withdrawal of your article, and this is how normally it will be watermarked and this will be put online actually, right? And there are also duplicated images could also be, or manipulated images could also be found out very easily. For example, here you could see this, all these images appears to be different, but understand all these images are the same image, right? So with a little amount of knowledge or on Photoshop actually, these images were manipulated, right? And same image has been presented actually, right? And again, plagiarism is also one of the important things, that is using others without others' ideas, or sometimes text without actually cite. So text should never be used as such actually, right? And sometimes they use for the ideas actually, they do not cite the authors actually, or acknowledge the authors actually. So this is what here you could see the paper that was detected, and here you could see almost the entire methodological sections actually has been reproduced by this from different paper actually, right? So again, you should always understand, right? So sometimes it is also very, very important to retain your data, original data actually, what you have collected during the experiments. For example, this is a paper that has been published in Nature actually, so it is also one of the highly cited paper actually, and it was ranked among the top 1% of papers that was published in the particular field, that is actually cancer science actually, in the last 10 years. But what problem was that when there was a problem with the image actually, so the original data for the image was not traceable. So ultimately, it resulted in the what we call the retraction of the paper actually. So understand, it is not very important to publish actually, but also retain the data. So today we have a large number of data repositories actually. So where you can deposit all your data actually, your raw data could be deposited, your images could be deposited, or anything could be deposited. So automatically what happens is that because in many of our institutions we don't have what facilities to retain the data actually. So that is one of the biggest problems. And now actually there are several data banks are available actually, where you can submit almost any type of data, and in future for future references actually, right? And this is what I was telling you about this fabricated review process actually. So what happens is that a person creates false email IDs and on actually names of a very well-known persons of that particular field actually. So automatically when he submits the paper comes to review for himself actually, and automatically he reviews his paper and submits a positive comments to the for the publication of that. So this is one of the examples to show you almost this particular person has actually published 9 papers actually in top rated journals like Chemosphere, crop production, right? And the total size of the total environment and all those things actually. Understand, sometimes when your time is bad actually, you can land in problems. Understand, when this person has submitted this what we call it as the list of reviewers to this crop protection. So this crop protection actually has this editor of this crop protection knew many of these reviewers in person actually. You understand? So when he was looking at this reviewer panel actually and their email ID, he got a suspicion, and on verification he found none of these email IDs which was provided by this author was actually true actually. You understand? So he has created email IDs of those people actually by himself actually, and he was doing all those things, and on inquiry it was found almost a large number of papers that were already published by this person was done on the same basis actually. So it has resulted in the retraction of many of the papers actually. And next comes a very interesting topic, that is actually the authorship center. So there is a large number of such funny cartoons available in the web actually. You might go through it, and many of the things you find it very funny actually. And on the extreme right actually, you could see one of the things which generally says that, right? So one of the co-author of the published paper was a night version, because understand who should be given an authorship is always a very, very important thing you should always understand actually, right? So it is now becoming a very complicated issue, right? So because the number of people who are involved in a particular study is generally increasing, right? And the second one is that actually the citations are very, very important. For example, getting grants or generally getting a position is also, or improving your position is also dependent upon many of these factors actually, right? So understand one important thing is that an author should have made a significant involvement or contribution to the design, data collection, or data analysis or interpretation, as I already told you, right? Or he should be involved in the drafting or revising the manuscript, or he should be involved in the final upload of the final version of the manuscript for publication, or responsibility of accuracy, integrity of all the aspects of research actually. Right? So one important thing or one line thing is what we could say is that he should have made substantial intellectual contribution to that particular study. Right? So when such type of things never happen, normally there are different types of things, that is what we call the honorary authorship. So in honorary authorship, what is done is that, so what they include a person who is well known in that particular field for improving the chances of getting the papers being published actually. So that is what we call it as a honorary authorship. And second one is what we call the gift authorship, as I already told you. This is one of the Asian culture, that is generally in Asian countries it generally happens. This is what we call the gift authorship. And last one is called as the coercive authorship, that is the forceful guest authorship, because certain people actually use their power. For example, I told the head of the institution or the head of the department actually, they use their power actually to actually get the authorship. So all these things are what we call it as the, these are the problems with the authorship certificate. To understand one important thing is that sometimes retraction could also happen, right? When you publish your paper without the co-authors. And so some people without the knowledge of the co-authors actually they used to include their names actually. So that should never be done actually, because that could also result in the retraction or withdrawal. So fixing the breach of ethics is not a very simple process. So it is a very long process. What happens is that the person, whenever an editor gets an information that there is a certain scientific misconduct of a particular study that has been published actually, so immediately what he does is that he goes through the manuscript of the publication, and he also seeks the help of the other editorial board members regarding this actually. So when they found that there is a certain problem actually with the paper actually, so they contact the corresponding author actually. So the corresponding author is almost a permanent person that is in any institutions actually, right? So automatically they seek the response of the author actually, the corresponding author. So when they don't receive any response from the corresponding author, they contact the other co-authors of the particular paper actually. So it is very easy today to find the co-author's email ID and other aspects actually, because they of the internet facilities that are generally available. So understand, when all these co-authors even they don't respond actually to the thing that has been or the claim that has been asked by the editor, then the editor contacts the institution of the place from where the publication can be made. You understand? So in certain institutions, what they do is that they used to set up the internal committee to look into the matter, and they used to submit the report to the editor actually. So in many cases, what they do is that the institution also does not take this much seriously actually. So one thing what they do is that they go on reminding the institution for every 6 months or 3 months actually, for certain period of time, right? So this is done only in the certain cases where actually the misconduct is not very evident actually. So it is not very evident. So when it is evident actually, what happens is that the editor himself actually takes a decision to retract the paper. So this is what is generally being done, and it is not a very simple process, it is a very long process. So this is for all those things actually, for your written publication, or it could be for a figure image things and all those things. And whenever there is a retraction actually, there are certain things that should be generally followed. So only under certain cases retraction should be followed actually. So there should be a clear evidence that the findings of the study are generally unreliable, right? And where there are major errors due to the miscalculation or experimental error, or the result of fabrication of the data, or falsification of the data. So falsification is generally image manipulation, right? And plagiarism. So plagiarism is very easy to identify today, because we have a large amount of software, and sometimes redacted publications actually, right? So they also sometimes contain materials without the authorization. Again, that could also be called copyright infringement actually, because you tend to take almost what we think is that publishing our own figures in the next paper is not a wrong thing actually, we always think, because already we have signed a copyright with the previous publisher actually. So whenever we want to reproduce it, automatically the permission has to be sought from the previous publisher for the use of the images, tables, or any thing that has to be republished actually. And again, certain things is unethical research, for example, without getting dual permission for animal experiments and other things, or human experiments actually. So this is considered to be analytical research actually, right? So again, published solely on the basis of what we call as the compromised or manipulated peer review process. Already I told you, right? So again, disclosing this major competing interest is also very important. So understand retraction notices also, for example, publishing or retracting the article also. There are certain important things that should be followed actually, right? Again, it should be done before much of the damage has been done, for example. For example, when it takes a very long time, automatically the paper gets cited for a very long time in the literature actually, so automatically it gets hundreds of the citations, or it's being cited hundreds of times actually. So that should not be done actually. So before, when the right time actually, this retraction notices or retraction should be done actually, right? Or sometimes our retraction is not is not necessary actually. For example, when the what we call the mistakes are very minimal, right? Automatically, when it does not affect the major results or the outcome of the study, the retraction is not necessary. For example, if you take up this example, what this person has done, that so this person has wrongly pasted the same image twice actually. You understand? So this has nothing to do with the results, what they are, the conclusions of the study actually. So automatically, what has been done is that they are just the correction could be published actually, or correction things could be published. So this also happens in certain cases. For example, people tend to miss the units of their parameters actually to be mentioned actually, and all these things actually could be corrected in your publishing the corrections in subsequent issues actually, right? So there are some studies on this. This I do not want to go into because already I have taken much of your time actually. So this is actually the retraction that was conducted in some African studies actually. What they have done is that, so the major reasons in Africa for retraction is one important thing is plagiarism, and second one is actually what we call the duplicate publication or duplication, right, of data, and third one is actually the error in data analysis. Are the three main factors actually, right? So for the Asian or the other regions actually, such studies are not available, right? And there is also another important thing, that is what we call it as the conflict of interest actually. Understand in certain cases what happens is that we don't understand what is conflict of interest is, because understand in certain cases what happens is that we tend to publish certain results actually, so which could affect or which could be far from true actually. You understand? So, for example, if you see there was a large surge in studies when there was a problem on the GM crops or genetically modified crops actually. You understand? And understand many of these studies were actually funded by companies which were producing GM crops actually. So automatically you could see, so this could not be unbiased studies actually. So automatically many of these studies actually tend to have certain type of biases actually. So this is what we call it as a conflict of interest actually. And sometimes this conflict of interest could also be due to personal problems actually, right? Or this could be understand this could be sure it's not always one of the important criteria for rejection of the paper actually. So that is why whenever you submit your paper to some standard journals, normally there will be the two criteria for the reviews that will be asked: preferred reviewers and non-preferred reviewers actually. You understand? So it is always here actually you would show who the other persons should not be studying your paper or studying your work actually. You understand? Because understand there are certain people actually who tend to publish that the data which comes for them for reviewing actually. So that is also one of the major, because I did not include that here actually because that is not the most concerned one regarding this topic, but this is also done in several cases actually, right? And the next important thing is the quality of the publications when you see. So today we have a large number of journals predatory, that is what we call it as the predatory journals actually. So because many journals have went online, so it has become a large business actually. So again, you should always understand that actually publication is a multi-billion-dollar industry, and this is one of the biggest problem actually. So there are many journals actually which publish your papers as they are submitted actually. So today night you submit your paper and pay the what we call the processing charge or whatever you say, page charge and anything actually, and tomorrow morning your paper gets published actually. Understand? So there are a large number of publications, right? So it is not only certain institutes are very less known institutes, certain well-known institutes, national university institutes as well as the big universities also actually publishing such type of journals, right? And here I will show you some of the examples how actually the journals tends to manipulate the impact factor and other things, actually, right? So this was this is actually the impact factor of the journal for 2008 to 2018. So in the 2014 actually the impact factor of the journal was something around 3.3 plus actually, and suddenly in 2015 it dropped to almost 3. Around 3 actually. So because understand this is a very multi-million-dollar industry, actually $1 trillion industry, the people are ready. So what they are seeing is that, so you know what how the impact factor is calculated. It is actually the number of papers published in the last 2 years divided by the number of papers cited in the last 2 years divided by the number of papers published in the last 2 years. Actually, that is how actually it is calculated. So on seeing what they found is that, so the number of papers or the citations were the same actually in the 2014. One thing was that there was large number of papers that was being published in that 2015. So this has brought down the impact factor. So immediately what the editor did was that he circulated a circular to all the reviewers automatically to reject the papers actually, to bring down the papers that is being accepted. So understand, this journal already has a rejection rate of more than 80% actually, and now this journal has a rejection rate of almost 95% actually. And you could see suddenly after this reduction in the number of papers that is being published, automatically the impact factor has jumped to great levels, and now it is actually in 2020 it is around 5.5 plus actually, 5.5 plus actually. So here you could see how actually scientifically these publishers actually manipulate these impact factors actually. And again, this impact factor manipulation is also done by, to a large extent by self-citations actually, right? So what we could see large number of publicated journals from economics, sociology, right, psychology or multiple business actually, they tend to actually improve their impact factor through self-citations actually. And here you could see in the SCA a large number of journals are withdrawn because of self-citations actually, right? It is not only self-citation. Sometimes what happens is that individuals also do the same thing to improve their H index, because H index now actually it was actually brought for the library people actually, but scientists generally took it as the criteria to show their credits actually. So what happened was that there are certain people actually, right, get large amount of this or improve their H index of their self-citations by themselves or their group actually. You could hear me, actually you could see the screen. Yeah, you could see the screen. Okay. So here what happened was that actually this person actually, he was an Indian person, actually you could see most of his H index was from his self-citation. Almost 95% of his citations are from his own paper that has been cited by himself actually. So there is also another study that was approved in actually in Spain, Italy actually, where in 2017 when the government started to have this H index as one of the main criteria in the promotions and other things actually, automatically the number of self-citations by the scientists has generally gone up. So I have given you a simple example of this particular person actually. So you see, if you take up this author H index of this particular author, right? So it is around 104. It is a very, very impressive, because anything above 30 is considered to be a great one, and here India got around 104 actually. So one simple thing is that just you remove the self-citation, you could see that his H index has gone down to almost 36. So there is a 65% reduction in what we call it the H index. So this clearly shows the amount of, because understand, so increasing your citations by yourself or your colleagues actually, by asking them to cite actually. So that is one of the biggest problem to improve your efficient H index or efficient index that is happening today in science. So here you could clearly see how a person has improved his H index. So see from 104 actually it has gone down to 36. This is 36 is the original still it is very impressive, but here you could see he has manipulated it to 104 actually. Right, and this has resulted in a large number of countries taking up a large number of measures actually. For example, if you take up China, so previously China was giving rewards, cash rewards, as well as promotions to persons who, whenever you publish in very good journals actually, right? So whenever you publish in impact factor journals or SEI journals actually, you are given cash credits actually, right? And now, because understand, so you could see here from 2010 where there was an annual publication of something around 13 or 1.3 lakhs actually, right? Almost it has jumped to almost in 2019 it is almost 4.5 lakhs plus papers is being published from China. So automatically you could see large amount of this actually manipulations is done by these Chinese researchers to improve their career actually and to get cash rewards actually. So there tends to be a large amount of fraudulent papers coming from China actually, right? So because many of these papers also containing duplicated images, right? And also dual submission, double publication, and all these things actually happen. So that is why actually now China has stopped providing cash rewards for publishing papers in science actually, right? And many of the guidelines as today has been given by many of the publishing companies actually, right? So try not to deviate from the original work. So whenever you are citing, you always use citation from the original work. Don't use the review or don't cite the reviews actually, and do not cite the sources which you are not read. So this is generally one of the important things, right? So it is not only that, do not cite your own paper or other friend's paper, right? Or the institutions or publications just to improve the citations, right? So you should also never use the number of citations for one particular point, because in some certain cases you could see in a journal for a particular point there will be 5 or 6 citations will be used. That should never be used, right? So understand it should always be undergone which the citations of the papers that is being referred to or cited should always be undergone the pre-review process. And now comes the question, who needs actually the training in research ethics, right? So whenever you ask anybody, the immediate question, answer that comes from their mouth is that I don't need it, but it is the other person who always needs it actually. So this is generally not true actually. So one important thing is the majority of the scientists or the researchers, right, in India or any developing country does not have a formal training in research. So this is one of the major drawbacks in our educational system actually, right? So again, students are also poorly mentored in the research ethics actually, right? So again, because this happens because the faculty also was trained in the same method by themselves actually, because they also failed to have this ethical education, right? So one important thing we should always remember is that all practicing researchers, right, need to practice research ethics, right? So when it is a good science plus good ethics, it becomes a good research. I think that is all from now from me. If you have any questions, I am ready to answer. Thank you, sir.
Thank you, sir. Interactive session. Please ask your questions.
Participants, if there are any questions you can ask. You can pose your question in the chat box also.
I kindly request Dr. S. Sivasankara Narayani ma'am to give the vote of thanks for this lecture.
Good evening all. It gives me great pleasure to propose vote of thanks on this important day for First, I would like to thank our principal for being the backbone in our college as well as department activities. Next, I would like to thank our chief guest, Dr. T. Muthukumar, for his excellent speech in scientific ethics and research publications. Through his speech, it was clear that what is a thesis and what is a dissertation and how to interpret the results and how to do research in a correct manner. As this goes, so a man without ethics is a wild beast used upon this world. So a set of moral principles and values are important in our life. So not only in our life, it is also required for our research also. The honesty and integrity in all the stages of the scientific research is very important one. So through his speech, it was very well understood. And I would like to thank our HOD of our department. His planning strategy always brings success to our brand endeavor. And I'd like to thank Dr. T. Sivakumar, organizing secretary, and other teaching faculty of the department. And I would like to thank the students who are the pillars of our college, society, and the nation. We deserve our appreciation and thanks for their cooperation and their enthusiasm to learn new things. Once again, I thank one and all. Thank you all. Thank you so much, sir, for your presentation.
Thank you, thank you, thank you.
Thank you, sir.
Now, participants being ready for poster presentation discussion. Abstract number 33. Jamia D. Peter S. Rajalakshmi K. Meena Gayatri.
Yes, ma'am. Rajalakshmi is here.
Lakshmi is here, okay. Video on. Video on. Video on. Participants, this is the time for memento distribution. I request our HOD Dr. T. Mohammed sir to distribute the memento to the judges. First, I call upon Dr. Mohammed sir, Assistant Professor, Department of Botany, to receive the memento from our HOD. Please, sir. Thank you, sir, for your judgment. Next, I call upon Dr. J. Pandya sir, Assistant Professor, Department of Biotechnology, to receive the memento from our HOD. Thank you, sir. Now I call upon Dr. Sivakasi sir, Assistant Professor, Department of Zoology, to receive the memento. Thank you, sir, for spending your valuable time in judging all our 32 posters. Thank you all. Now it's time for discussion forum in poster presentation. Rajalakshmi, be ready.
Yes, ma'am.
Good evening. Okay,
Good evening, ma'am.
Your first question. So, have you characterized the species? So, intended for the dedication process to the molecular or any of the biochemical characterizations?
Yeah, ma'am. Actually, we performed molecular and biochemical characterizations, but we got the result of molecular characterizations just day before, so I wasn't able to include it in that. And in biochemical characterizations, all the organisms are gram positive, and two organisms are bacillus and two are cocci, ma'am.
So what is the organism you have find out?
Yeah, ma'am. One is we have sequenced only three organisms which are effective, of which one is Streptococcus species, and the other two is Laciniobacillus, ma'am, of different species. Just have to check the result, ma'am.
Okay, actually, is there any specific reason for selecting the temperature of 37 degrees Celsius and other than 20, 30, 40 and 50 degrees Celsius?
Yeah, ma'am. We got a better growth at 37 degrees centigrade and the better degradation at 37 degrees centigrade when compared to other 20 and 50 degrees centigrade. So we found that optimum, and now we are working on to enhance the degradation of the paracetamol using that particular temperature, ma'am.
Okay, we want got the results right and any of the specific reason we can get. So what is the mechanism act on that 37 degrees Celsius?
Sorry, ma'am, I don't get you.
We have found that 37 degrees Celsius is optimum for that organism. So what
Yeah,
is
ma'am.
the reason for that? So why does
Yeah,
not?
ma'am. We performed optimization studies for 3 degrees, which are commonly reported. So we performed for a lesser degree, room temperature, and as well as for the higher degree. We got growth in all the three, but maximum growth was observed only in 37, and maximum degradation was also observed in 37. So that was the optimum temperature in which we can get a better result and go for further studies. So we chose 37 degrees centigrade for future studies as well.
Okay, okay. Next question. So
Yes,
why
ma'am.
did you select the paracetamol in your studies?
Yeah, ma'am. So actually, we collected soil samples from the dump site. So when we are going and collecting the soil sample from the dump site, we saw many medicine wraps which are available there. So most of the medicine wraps which are commonly used by the public is paracetamol. So that is the main focus that we chose the paracetamol as a compound of degradation.
Okay, so do you know what is the self-life range for the degradation of this paracetamol?
Sorry, ma'am.
What is the self-life range for the degradation of the paracetamol?
Actually, the range of degradation we used 100 ppm to 500 ppm, and we got better results for 100 ppm, but the other organism which showed better results at all the 4 ppms. So we are now further scrutinizing the organisms, and we are working on it furtherly, ma'am.
Okay, good. I am asking the normal degradation values. Okay, you are
It's
working
60. It's 67%, 75%, and 77%, ma'am.
okay, good. Okay,
Thank you, ma'am. Thank you so much, ma'am.
Next abstract number 36, Satyabhama Lakshmana Narayanan,
Yes,
Subhadevi. Please, sir, Subhadevi. Sir, Satyabhama is here. Okay, Satyabhama, your abstract number 36.
Yes, ma'am.
Okay, good afternoon.
Good afternoon, ma'am.
Okay, are the values mentioned in this graph or statistically different or not?
Ma'am, once again ask, ma'am.
Actually, are the values mentioned in this graph are statistically different or not? Especially between the standard and the sample.
Ma'am, we are characterized by the UV visible spectrum analysis, ma'am. Then it also is analyzed by the HPLC analysis, ma'am. In biochemical properties, we analyze only standard deviation method, ma'am.
Oh, okay, okay, okay. So what is the quantification? How will you extract that?
Ma'am, I quantify the total carbohydrate, total protein, and also in lipid. Then we are also in extract the biochemical protein from the Halimita thallus, ma'am.
Okay, what is the mg of the phycobilic proteins from per gram of serum?
It's in milligram of 1 milligram, ma'am. 1 milligram of sample is taken, then evaluate the pigment profile.
Okay, ma'am. Okay, thank you.
Thank you so much.
The next participant, Uday Ramkumar, abstract number 37. Are you here? Uday Ramkumar. Okay. Next, Jayaprabha. C. Jayaprabha, abstract number 39. Next, Naveen Kumar, Maheshwari, Mahendran, Srinivasan. Naveen Kumar, are you ready? Okay, Naveen Kumar is here for discussion.
Number 1, abstract number 41.
What is the conclusion of your research? And tell your objective of your work. So what is your objective of your work?
I choose the plan is my plan is in 3 instructions. It is a waste plant, so the plant is using the antimicrobial activity.
Thank you.
Okay, next participant, Soundari, abstract number 43. Soundari, are you here? Soundari. Okay, next participant, Lokesh, abstract number 45. Lokesh.
Ma'am, I'm here, ma'am. Yes, ma'am, I'm here, ma'am.
Okay, you just get the question.
45.
Good afternoon. Actually, my first question is phycyanine a toxic compound?
No, ma'am, it's not a toxic compound, ma'am.
Okay, you have used against the cell lines, right? Which type of cell lines have you used for the study?
It's colon cancer cell line, ma'am. HT2 line, ma'am.
Okay, it showed the activity against the colon cancer cell line. So have you tested against the normal cell line for this?
Normal cell line, I have not tested it, ma'am.
Okay. So how can we use this for a full food colorant?
Yes, ma'am, we can use as a food colorant, ma'am. By
Other
extracting, ma'am, by extracting the phycyanine pigment, and then by drying them, and then we can use as alternate for the food colorant, ma'am.
Okay. So what is the IC50 concentration against the colon cancer cell line?
Yes, ma'am. It's 45.29 microgram per ml, ma'am.
Okay. Have you optimized the extraction of the phycyanine, right? Which media produces the more amount of the pigment and why?
Yes, ma'am. Nutrient broth produces a more amount of pigment compared to Kingsley medium, ma'am. Because the Pseudomonas grows well in nutrient broth with a sufficient nutrients in it, ma'am. Mainly, it needs a carbon first to grow, ma'am.
Okay, thank you.
Yes, ma'am. Thank you, ma'am.
Next number 48.
Save Malada, Here, abstract number 48, Devalada. Next, abstract number 49, Ajit Prakash. Abstract number 49, Ajit Prakash. No. Next, abstract number 51, Esther Rajalakshmi. Abstract number 51, Esther Rajalakshmi. Then abstract number B1, Muthukumar. B1, Muthukumar. Next, B2, Shiva. B2, Shiva, are you here?
Yes, ma'am. Good afternoon, ma'am.
Shiva, are you here? Yes. Okay,
Yes,
get the question.
Okay. So your question is, what is the mechanism behind the degradation of waste?
Ma'am, can you please repeat your voice breaking, ma'am?
What is the mechanism behind the degradation of waste?
Ma'am, there are so many mechanisms in degradation of waste and plastics, ma'am. In waste, there are two types, that biodegradable and non-biodegradable, ma'am. In biodegradable, we use microbes like bacteria, fungi, ma'am. But in non-biodegradable waste, there is no mechanisms, ma'am. So we are doing a project in a synthetic and non-synthetic wastes, ma'am. So in a future aspect, we want to delight us. So that's why we using this project now, ma'am.
So
But we
what are
do
the okay? So what are the microorganisms involved in the degradation purpose?
Ma'am, can you please repeat, ma'am?
So what are all the microorganisms involved in the degradation process?
Ma'am, we are bacteria and fungi, ma'am. In bacteria, we use Bacillus, Corynebacterium, Staphylococcus, Streptococcus, etc., ma'am.
Okay, ma'am. Thank you.
Thank you so much, ma'am. And thank you for this opportunity, ma'am.
B3 Padmashree.
Yes, ma'am. Good evening, ma'am.
Okay, good evening.
Yes, ma'am.
So your question is, what is cellulosomic approach?
Ma'am, in plastic, the microbial biodegradation and attaching the plastics and microbes is complicated, ma'am, because of the hydrophobic effects of plastics, ma'am. We
Okay.
using enzymes, ma'am, extracted from the bacterial strains, ma'am.
Okay.
By using the enzyme from bacteria, the cellulosomic approaches is done, ma'am.
Okay, actually there is a large number of plastic waste are there in our world. So it is possible to remove with the help of the tiny organism?
Ma'am, we are researching in some plastics only, ma'am. In PET and polyethylene, ma'am. More research is done in polystyrene, ma'am.
Okay. Thank you. Good.
Thank you, ma'am. Thank you for the opportunity, ma'am.
Next to B4, Mary Isabella Fanani.
Yes, ma'am.
B4. Your question is, how will
Yes, ma'am.
how will your work is useful to the society?
Ma'am, can you repeat, ma'am?
How will your work is useful to the society?
Ma'am, nowadays usage is very much higher, ma'am. Like you use chemical to the soil and you create soil infertility as well as pollution to the water table. When you biology way approach like biofertilizers to apply to the plant instead of chemicals, it will be useful to the plant as well as we won't manage the soil fertility and soil profile and as well as not contaminating the water table.
Which organism used in your study?
We used Pseudomonas.
Okay, where did you isolate that organism?
We isolated from the rhizospheric soil, ma'am, from the plant itself. These are indigenous organisms which are already present. We scrutinized and made them very efficient for the solubilization of zinc, silicate, and selenium. So, why we concentrated on these three is because majorly many researchers are concentrated on macronutrients like nitrogen, phosphorus, and potassium, and where minimal concentration is shown on the micronutrients such as zinc and silicate and selenium. So that is why our interest was on the micronutrients rather than on macronutrients.
Actually, what is your main objective of your work?
We wanted to isolate organisms which could do all three actions, like the zinc, selenium, and silicate. And further, we should further studies are going on, like formulating this organism as a biofertilizer and implementing it to the plant. And we also observe growth on plants. The studies are going on with tomato and chili. The research we haven't arrived on the research. That is the reason I have put it here.
Good. Thank you.
Thank you, ma'am.
Number 8. Number 8, Pratibha. Abstract number 8, Pratibha. Abstract number 16, Subha Devi.
Yes, ma'am. Good evening, ma'am.
Good evening, ma'am.
Yes, ma'am.
Your question is, so how to quantify the microbial proteins in your studies?
What, ma'am? Tell me, ma'am. Ma'am, your voice is breaking, ma'am.
Yeah, how to quantify the microbial proteins in your studies?
In my studies. Ma'am, phycobility proteins is a family light harvesting pigment, ma'am. I'm using my studies extraction and estimation of phycobility proteins in kappa picus sulfurily, ma'am. And proteins, lipids, carbohydrate, and also antioxidant studies, ma'am.
I'm asking actually how much MG you are getting in your extraction, particularly the phycobility proteins.
Take 1 gram of sample, ma'am. 1 gram of algae sample was taken, ma'am.
Okay, thank you.
Okay, ma'am. Thank you, ma'am.
29, Akhilesh.
Yes, ma'am. Yes, ma'am.
Akhilesh, your question. So, did you check the nutrient content of the compost?
No, ma'am.
So, what is your main objective of your work?
Ma'am, biodegradation of sugar factory waste and paper waste and fruit peels, ma'am.
Okay. So what are you going to say about this work?
Ma'am, this vermicompost is improve soil aeration, ma'am. Enriches soil with microorganism, improve water holding capacity, ma'am.
Okay.
To help balance the pH level of soil, ma'am.
Okay. And have you used this compost for any other purpose? Is it in process now?
Yes, ma'am.
Okay, but thank you.
Thank you.
Good evening.
Albino wings. Albino wings.
என்ன கேட்டானு தெரியல.
Students, any other participants are there? Students, any participants are there? If you want to answer, you can answer now.
The participants of previous day posters also can join the discussion forum today. Nishant, are you here? Nishant,
Yes, ma'am.
are you ready for the discussion?
Yes, ma'am. I'm ready.
Where's the question?
Nishant, 7.
Okay, good afternoon. Your question is, so why snake venoms are chosen for our study? Nishant? Nishant?
Yes, ma'am.
Okay.
Ma'am, venoms are having a special activity which is spreading into our body very quickly. Their activity rate in our body is very high. For example, if a snake bite has happened, the venom or the protein which is responsible for the poisonous activity will transfer quickly to the nervous system and directly attack the brain, nervous system, immune system, or their activity rate is high.
Okay. Sure. Actually, is they safe to use as antiviral peptides?
Yes, ma'am. When you are using for your you
Sorry?
have to use the main when you are using a protein for purpose that has to have a I have chosen this for activity their activity. They have an antiviral activity that cost point for me for because I'm doing in COVID. I'm going for antibiotics.
Actually, Nishant, you have collected the receptor and ligand, right? How will you choose the receptor and ligands?
Ma'am, receptors are basic, ma'am. The common, the 2-GTP main protease of COVID-19 was reported as the basic for molecular docking and drug designing purpose by many scientists. And ligands I have chosen was my purpose, which is they having antiviral activities and also having the spreading activity in high rates.
You have chosen, right? Based on the other reports, you have chosen this particular receptors. Okay.
Yes.
Thank you.
Thank you, ma'am.
Any other participants? Today or previous day?
I kindly request Dr. K. Nalini ma'am to give the report of the seminar.
Respected Principal Dr. C. Ashok, dignified head of the department Dr. P. Mohanika, my dear faculty friends, all the participants from other institutes and from other college, a warm good evening to all. I am here to present the report of the DBT sponsored 2 days national seminar on emerging trends in microbiological research 2021. DBT has sponsored 16,000 for organizing the seminar in a virtual manner. So totally 215 participants are registered for this seminar, and we have received 61 abstracts from different institutions all over India. So we have received abstracts from New Delhi, Haryana, Pondicherry, and different regions of Tamil Nadu like Coimbatore, Thuthukudi, Chennai, and then Namakkal, different areas with another Sivakasi. The seminar started with a welcome address by Dr. P. Mohanika, sir, head of the Department of Microbiology, followed by the presidential address of our dignified Principal Dr. C. Ashok. He focused on the importance of the interdisciplinary research. Then the scientific session started. Totally we have 4 different scientific sessions with 6 lectures from eminent personalities. The first resource person of the session is Dr. S. Kannan. The session started with the introduction to the chief guest by Dr. K. Nalini, Assistant Professor, Department of Microbiology. And Dr. S. Kannan started his guest lecture. He is the head professor, Department of Zoology from Periyar University, Salem. He explained the technique Sivakasi technology in gene editing. He explained all the details of the enzymes and the applications of the techniques. The session 2 was followed by Dr. N. Ayya, Senior Scientist, Department of Biotechnology, CLRI, Chennai. He is an alumni of our institution. He made his session on synthetic biology through green fluorescent protein. He explained how to synthesize different types of synthetic proteins, and he also explained the various applications of the fluorescent protein in different biological fields. Next, the third session is by Dr. Dhansharan sir, Associate Professor, Department of Microbiology. He shared his research experience in probiotic foods and gut microbiome. He explained in detail about the importance of probiotics, prebiotics, symbiotics in maintaining gut microbiome and also in protecting and preventing from diseases. We also had a discussion about the new topic called psychobiotics, which plays a beneficial role on mental health and emotions. So after the first day session, the discussion forum of e-poster has been conducted. So after total 32 posters, 16 total posters were discussed on the first day session. Next, the second day session started with the scientific session by Dr. Vijay Kumar Velu, who is also an alumni. He is from Emory Vaccine Center, US. He shared his research contribution on HIV strategies. He focused on the importance of CD4, CD8, and the natural killer cells. He also stated the importance of antiretroviral therapy, and he focused more on the viral reservoirs. He shared his research work on the virus infected rhesus monkeys and post-virus infections, role of interleukin-15 cytokines in treatment of the HIV. And the next session was by Dr. S. Velayatha Prabhu, Assistant Professor, Department of Biotechnology, Bharat University. He discussed about the need of animal model in research, the importance of rodents and non-rodents fish in different research. He clearly clarified the confusions behind the selection of animal models available based on their lifespan, reproductive rate. Through his session, all the participants got the answers for the question why to use, where to use, and how to use animal models. The participants came aware of different types of systemic and organ-specific autoimmune diseases. He particularly focused his research work on anti-positive lipid syndrome. Next, the sixth session was presented by Dr. T. Muthukumar, Associate Professor, Department of Botany, Bharatiya University. He explained in detail about the thesis writing and ethics to be followed in publishing research work. And the session was followed by a discussion forum on e-poster presentation. Nearly 17 posters were being discussed on the second day. So out of 32 posters, all the participants from different institutes have presented well and they made a discussion forum. So out of these 3 best posters will be awarded with prizes and certificate. And in addition to the best posters, 3 best interactors award has been also selected who interacted well throughout the session. The session came to end with a vote of thanks and national anthem. And thank you for giving me a chance to present the report of this seminar. Thank you all. I request any one of the participants to share their feedback regarding these 2 days seminar. Any one of the participants can share their views. Lokesh.
Yes, ma'am. Good evening, ma'am.
Good evening, Lokesh. You just share your views on this seminar.
Yes, ma'am. The session was a very informative one, ma'am. Especially the today's afternoon. He told how to publish in journals and what are the things we can publish in 2 journals at a time, or how we can do. He told, ma'am, this was very informative one.
Thank you, Lokesh. Any other participants?
Thank you, ma'am.
Lokesh, introduce yourself. You are from?
Yes, ma'am. I'm a student of you, ma'am. Now
you are not audible. Lokesh?
Yes, ma'am. I'm a student of you, ma'am. And now I'm doing PG in Vellore Institute of Technology, ma'am. PG first year, ma'am.
Okay. Next, any others?
Savina.
Sabina. Okay, Sabina,
I'm Savina Solanki. I like the whole seminar about the. I'm sorry, can you hear me? Can you hear me?
So your voice is not audible. Can you make it clear audio?
I'm not sure my voice is going or not. I'm Savina Solanki. I have attended the whole seminar on emerging trends in microbiological research, and I really like today's afternoon session by Dr. T. Muthukumar. I really like the whole how to write or the scientific ethics in research publication, which is really good to know about all the aspects which I don't know before that. I'm one of the participate for the e-posters from Delhi, University of Information and Engineering Technology from Delhi. I find the whole seminar very good, and I really like the animal models and how to work on the animal models in yesterday's lecture too. Thank you so much.
Okay, thank you for that. Okay, next is the time for announcing the best poster presentation. We have selected 3 posters. It is a difficult task for the judgment because most of the posters are presented in a very manner. The discussion forum is also very nice. So we have selected based on the 3 judges and the discussion forum. We have selected 3 best posters for this national seminar. The best poster award goes to S. Rajalakshmi, abstract number 33. He She is from Stella Maris College, Chennai. Radhalakshmi, are you here? Radhalakshmi.
Thank you so much, ma'am.
Okay, congratulations, Radhalakshmi.
Thank you so much, ma'am.
And the next best poster award goes to D. Diksha, abstract number 26 of Authorship University, Coimbatore. Diksha, are you here? Diksha? I think she is not here. Okay, congratulations, Diksha. And the next poster, best poster award goes to Shifa Vanmati from Sadakapula College, abstract number 18. Shifa, I think she is also absent today. Okay, congratulations for all the participants and the winners. So you made the best e-posters. And the next best interactor award. So we have selected this based on the interactions. We have selected 2 participants, Deepak and Padmashri Krishna Kumar. Deepak and Padmashri Krishna Kumar. Okay, Deepak and Padmashri Krishna Kumar are receiving Best Interactor Award. And the next Best Interactor Award goes to Vimal Raja of 3rd B.Sc Microbiology, Ayya College, and Shakti Priya of 2nd M.Sc Microbiology, Ayya College. Congratulations on all the poster Best Interactors. Thank you, participants. Okay, the participants, you will receive the certificates in next 2 weeks. Okay, and best poster award will be posted in courier, and the participation certificate and best poster certificates all will be available as e-certificate, and we will send them to mail in next 2 weeks. Thank you, participants, for your patience learning. Now it is a time for vote of thanks. Now I request Dr. Sivakumar, Assistant Professor, Department of Microbiology, who is the organizing secretary for this 2 days national seminar, to propose vote of thanks. Please, sir.
For this historical event to all dignitaries assembled to our days.
അവര് വരുമ്പോതും ഒരു ക്യാമറയോടെ വന്നിനോ വരുമ്പോൾ നിറയെ പേര് മാസ്ക് പോടല്ലേ എല്ലാരെയും ഫോട്ടോ എടുത്താവ
Platform for participating and contributed to the national seminar on emerging trends related to Microbiology 2021. First, I thank EBT New Delhi for providing funds to conduct the seminar. Next, I thank our dynamic personality and correspondent Guru G. B. Aravith sir, who, despite his busy schedule, has found time to grace the seminar to make grand success. On behalf of our department of microbiology, we are very happy, grateful to our college principal, Dr. Sivakasi, for his words of encouragement, his able guidance, and continuous support to our department. Then I thank our head of the department, Dr. B. sir. I feel grateful to thank our head of the department for his support, vision, and commitment to design this national seminar with successful. It is my job, or my job is meaningless if I am not mentioned to such people. I would like to thank Dr. Ismail sir for sharing the sir, associate professor, the head professor for the department of zoology for sharing his wonderful thoughts, provoking information on the topic related to the CRISPR gene editing technology. Next time, my heartfelt thanks to the honorable speaker, Dr. Ayya, who delivered mind-blowing message on the topic entitled the discovery of fluorescent proteins. Next, I place my record of gratitude to most respectful speaker, Dr. Kaleeswaran, who made his wonderful information on the topic to get my coping. Then I also express my heartfelt gratitude to Dr. Vijay for his valuable sharing on HABQ standards. I express my deep gratitude to Dr. S. to have made his session as the milestone to the participant on the topic entitled autoimmune disease, the medical model for autoimmune disease. Then I also express special gratitude to Dr. B. Muthukumar sir, who have shown new platform for easily publish our findings in our field. through his speech on scientific ethics and research and publications. That I may keep the thanks and appreciation on behalf of our department and college those who have contributed for e-posters. Next, my heartfelt thank to my colleagues for the cooperation in making the concern a resounding success. I heartfelt thank to Dr. Raniman, Dr. Mrs. P. Maheshwari, Mrs. Dr. Vijay Shatiman, Dr. S. Jason sir, Dr. S. Mahendran sir, Dr. B. John Wilson sir. Next, I thank to people of Visual Communication Department for the technical support rendered during the seminar, and I express my sincere thanks to the evaluators of the e-poster in PC students. Next, my heartfelt thank to young and energetic in expressing my gratitude to the students and the college participants of various colleges for extending kindness, interest, and the continued support. Once again, I thank you for all your attention. Thank you.
Now we are ending the session. Thank you participants for giving us the support in organizing this seminar. Thank you all. We can wind up now.
Thank you, ma'am.